Any questions on the review of this or the nomenclature here, how to draw out these molecules? Okay. I'm going to back out real quick because I actually don't remember what else I wanted to talk about today. So bear with me for a second. Spend 10 minutes talking about wikis. All right. Here's what we're going to do. Most of them look great. I don't have much of an issue with them at all. What I wanted us to do was actually to kind of spend time in groups of 2 or 3 reviewing these building blocks. And again, the main things that I want to get across are what's the name of the building block, what's the, and I guess what I'm looking for is like the simplest of names. So like an amino acid. For some people might refer to like the sugar molecule. They might say sugar, carbohydrate, glucose as the specific one. We'll just use generally monosaccharide to refer to these sugars. So give me the monomer name, give me the polymer name. One thing that I think got missed on some of these, what are some of the functional groups? So like a lot of individuals identify the amino on the amino acids, but there's another functional group there as well. So what is that other functional group? Okay, and then the last part, I want you to double check some of the answers on where do we find these. And I want to rephrase that question slightly. Where do we find these in prokaryotic cells? We are exclusively this semester going to focus on bacteria, with the exception of like 2 class periods for viruses. But because of that, we have to start thinking about, okay, so what's the difference here between prokaryotes and eukaryotes? Okay. What is one key distinction between the two? Presence or absence of a nucleus. So if, for example, an answer says, hey, the genetic material, you'll find it in a nucleus. Nuh-uh, not in this class. Okay? You will in eukaryotes, but not in prokaryotes. So take a look at that, work with somebody else, kind of go through the answers that you had. I will post up my original with these images on here, at least a little bit, so that you can see those and begin to get a feel for the different features of these. And then we'll spend, I think maybe we'll spend like 5 minutes in conversation, a couple of minutes just reviewing. And then we'll talk about specifics of these. All right. Where would we find this? Where would we find sugars? And it doesn't have to be glucose, but monosaccharides in general. Where do we find this in a bacterial cell? Okay, we'll save that for a later conversation. Last one. Simple, or what is this single one? Nucleotide. All right. Could be a ribonucleotide or could be a deoxyribonucleotide. The difference is based on the sugar. We'll come back to that way later. Some folks pointed out in their wikis that there are 3 parts to this. We've got the triphosphate or phosphate groups. We've got a ribose sugar base. And then we've got a nitrogenous base over here. Major functional groups in here that we're going to pay attention to: hydroxyl, amino, phosphate. Okay? And then this, of course, is used to build our DNA and RNA. So there are 4 functional groups that I am concerned with you knowing. They are carboxyl, amino, hydroxyl, and phosphate. What is true about all 4 of these? Or any one of them? So take a look at the bonding structures. What properties become apparent as we begin to take a look at the individual covalent bonds for each of these?
Can you repeat the question, please?
Absolutely. So what properties become apparent when we begin to look at the covalent bonds in these functional groups? So for example, between an oxygen and a hydrogen, what properties exist in that covalent bond? Or between a nitrogen and the two hydrogens? What's true about those covalent bonds? Go back to what we talked about on Wednesday. Shall we play a game at Hangman? This is always the fallback, okay? Hangman. All right. Damn it. All right. They're polar bonds, right? In other words, they've got partial positives and partial negative charges. They're reactive. So a lot of these ones with hydroxyl groups, with phosphates, which don't have partial charges, actually at neutral pH, these oxygens have full negative charges on them. They're reactive. And this is really important because that reactivity makes them it allows for them to be involved in reactions. One of the main reactions that each of these is involved in, at least when we're building from monosaccharides to polysaccharides, or from a monomer to a polymer, are dehydration reactions. So, what is a dehydration reaction? What was that? When you're removing water. Exactly. So the basic format that we follow when we look at these is if we've got two molecules, we'll just call them R1, and let's say this one's got an OH group on it, and then we've got R2 with another OH group, and they react together, they come together in an active site of an enzyme, and we want to dehydrate them to create a bond. We identify where the water is, and the product then is bonding these two molecules now into a single one. Every single one of these individual building blocks that we looked at follows this exact type of reaction scheme in order to be built into polymers. Okay? Let's walk through one other example. Let's take a look at an amino acid, or two amino acids. When amino acids come together, we always, based on how they line up in the ribosome, we always get a reaction between a carboxyl group and an amino group. So here's what I want you to do. Identify from this carboxyl group and from the amino group on the right-hand side, where are you getting the water from? So where are those two hydrogens and oxygens coming from to create water and then to create a bond between these two amino acids? Okay? Take a minute to at least identify the water first. All right, so where's our water coming from? The carboxyl group provides what to that water molecule? DOH. that means that the amino group has to provide one of those two hydrogens, right? So that becomes our water. What that means is the following. This bond here, broken. In other words, to complete the octets that are required for each of those, for that carbon and nitrogen, they have to reform, or they have to bond together. So the end result of this process then is our two amino acids bound together, where I'm just going to kind of draw this in red. Now instead of having water, you've got a carbon-nitrogen bond. This is known as a peptide bond, hence the name polypeptide. Okay, so the end form will look like this. That's our end product. Opposite of a dehydration reaction is what kind of reaction? Hydrolysis, water breaking. Okay, and in that case, what we'd get is water would go into that bond, break them into two individual amino acids. I'm not expecting you to be able to draw these by any means. What I am expecting you to be able to do is to identify dehydration and hydrolysis reactions. The importance of this. Dehydration reactions is an anabolic reaction. It's one that requires energy to take place. So typically we think of this as being, these reactions are used to build a cell or to build molecules. Catabolic reactions are ones that involve hydrolysis or breaking down. So for example, when we talk about a cell breaking down glucose later on to take that glucose molecule and produce ATP from it, that involves hydrolysis reactions, water breaking reactions, to break these into simpler molecules. All right. All right, let me get out of this drawing. Go back to slides for a few minutes. Okay. Probably the two most important structures to cover today are... apparently that does not want to load that way. Okay. I think that the two most important for us to cover today are probably phospholipids and lipids in general, and then also cover some of the basics of protein structure. Okay? So that's where we'll spend the last of our time today, and we'll see how far along we get with it. Biggest thing with phospholipids. Phospholipids, as we've said, these will be used to build the cell membrane. Okay? A phospholipid bilayer is two layers of phospholipids where these head groups, which are polar because they've got a phosphate on them, these are facing the aqueous solution, and then in between we've got the fatty acid tails. When I think of these, I think of an Oreo. Why? Because when you dunk an Oreo into milk, the chocolate, which is hydrophilic, gets all soggy and wet, all right? But the middle stays dry because it's nothing but fat. Really good fat, but nothing but fat, okay? So, Oreo. What this picture is trying to model here is the difference between what we would call saturated and unsaturated fatty acid tails. What is the distinction between the two of them? So what is a saturated fatty acid? Go ahead, Annie. Has maximum number of hydrogens. In other words, every single carbon is bound to two hydrogens and two carbons, with the exception of the one at the right at the end. Right. So this image on the left-hand side here would be a saturated fatty acid. Unsaturated, on the other hand, have a double bond somewhere within the hydrophobic tail. The types of saturated fatty acids that we see in nature, ones that aren't artificially created, are cis fatty acids. And cis means same here. What we mean by that, when we're talking about a cis fatty acid, is the following. So I'm just going to kind of zoom in on this little portion right here. So carbon, 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 carbon. Each of these is bound to at least one, if not more, hydrogens. Oh, oh no! My picture's gone. Damn it. Try that again. All right. When we say same here, we mean that the two carbons next to the double bond, the adjacent carbons, are on the same side of that double bond. Double bonds are rigid. They're not like single bonds where two atoms can easily rotate along that bond. Double bonds, they're kind of stuck into place. So for fatty acids that have a cis saturation or a cis unsaturation, they're always going to have this bit of kink or this bend right here. What does that do? How does that affect the properties of that fatty acid? And probably the simplest example is to think about oil versus butter. Butter is full of what kind of fatty acids? Saturated or unsaturated? Saturated. Saturated. At room temperature, butter is solid. Olive oil, on the other hand, lots of unsaturated fatty acids. And oil is fluid at room temperature. And so the difference in that cis unsaturated versus saturated fatty acid tail gives this property of fluidity. This is really important when we start to talk about bacterial membranes. Bacterial membranes have to maintain a consistent fluidity in order to maintain homeostasis. One of the ways that we can think about this is, let's say we've got in a typical cell, for example, E. coli. Let's say we've got a cell of E. coli and there we go. Let's say at 37 degrees Celsius, which is body temperature. Let's say that 90% of the fatty acids are saturated and 10% are unsaturated. And that percentage is required to maintain a consistent movability of that membrane. Now, what happens to butter when you stick it into the microwave, turn the microwave on? What happens? It melts. In other words, heat will take saturated fatty acids and end up increasing their movement, changing it from a solid to a liquid form. Right? And if you let it cool back down, it solidifies again. What's going to happen to a bacterial membrane then under the same sort of circumstances? What will happen to the membrane or the movement of that membrane as temperature goes from 37 to, say, 45 degrees Celsius? Does it move quicker or slower? It moves quicker, right? As it moves quicker, it allows for things to pass through the membrane much more easily. In other words, it disrupts cellular homeostasis. So how does the bacterial cell respond? What might a bacterial cell like E. coli do in order to restore homeostasis at that new temperature? I'll give you a minute to think about this one, and I'll repeat the question too. So again, the scenario here, we've got a bacterial cell at 37 degrees Celsius, 90% of the fatty acids in the phospholipid bilayer are saturated. Temperature goes up to 45, increases the movement of those fatty acids, increases permeability. How does the cell accommodate, or how does the cell change the composition of the phospholipid bilayer to restore homeostasis, bring down the speed of movement in that membrane. Okay. I'll give you a minute or two to think about this one. Feel free to discuss it with somebody else, and then we'll talk about it together. It's the fact that things are getting hit because of the mobility. So as mobility is going to increase, that's going to increase the likelihood that you're getting separation of those fatty acids, which allows for things for water, other typically non-polar molecules to get through that shouldn't be getting through. Does that have to do with your cycles, or is it more about? It's more about the fluidity. It's not so. Yeah. Oh.
Sorry, that was really confusing.
No, that's okay.
Actually a question.
Okay, let's walk through this one together. So temperature increases. We've defined that as that temperature increases, we'll see an increase in movement or mobility of those phospholipids. So they're going from, I shall model speed, they're going from, if we're looking at the top down, movement like this at 37. To now at 45, they're going much faster, okay? And that disrupts, that will create the opportunity for polar molecules, like water, for example, to actually get into the cell or to get out of the cell much more easily than they are at 37 degrees Celsius. How does the cell compensate for that? What might it do? How can it slow down movement of those phospholipids? I got an idea. Everybody stand up real quick. And we're going to locate over into this area. Okay? Okay, we're all going to be phospholipids with one saturated fatty acid and one unsaturated fatty acid. So choose your left or right arm, whichever one you want to, and that becomes unsaturated. Okay, so everybody stick out one of your arms. Come on in, we're all going to try to fit into this area right here. Okay, come on. Don't be afraid. And you can't touch anybody else. Did we all fit? No. Okay. So how do we get ourselves to fit? Put your arms down. Now try to fit. Like an elevator, right? People outside are going to be like, what the hell's going on? Okay. So how does the cell compensate to bring these fatty acids or these phospholipids closer together? Doing that will decrease permeability, right? How does it do it? that breaks it apart. What did we do? We put our arms out. We changed our shape. We went from being one saturated and one unsaturated to now being what? Both saturated. That's how a cell compensates for it. Increase the percentage of saturated fatty acids. Therefore, it allows for those those phospholipids to group tighter together, decreasing that movement. Okay? I mean, obviously, this way we will not be able to move around, but if we all stuck our arms out and spread out again, it'd be much easier to move. So that's how cells accommodate for those. All right. Questions in the small group? All right, you can go sit back down. So there are some exquisite strategies to deal with this. There are some microorganisms, not bacteria, but what are archaea, we call them extremophiles. They can live in temperatures of boiling water. So 212 degrees Fahrenheit, 100 degrees Celsius. The way that they deal with this issue is a little bit different. Instead of having a phospholipid bilayer, they actually have a phospholipid monolayer, where the it's got two polar groups and then phospholipids connecting those two. And that actually reduces movability even more, so it allows them to accommodate with those really high temperatures. So there's not one strategy. But the goal of these strategies is to maintain homeostasis by playing with the percentages or concentrations of phospholipids and fatty acids. All right. I think I'll just spend a really quick minute discussing amino acids because I want to make sure that we have time to kind of cover a couple of basic things. Amino acids, the basic sequence of amino acids is known as the primary sequence. Okay? A lot of times, and we'll do this a little bit in class 2, is analyze the original amino acid sequence, but that only tells us part of the story. Really, we don't really understand much about proteins until we can see the three-dimensional structure. And that three-dimensional structure is created by intramolecular interactions. Interactions that will bring two ends of the protein together or to repel them from each other. So we have secondary structures which are known as alpha helices, beta sheets. These are hydrogen bonds that form between amino and carboxyl groups of that main amino acid structure. The side chains contribute to what we call the tertiary structure, or that global globular formation of a protein. And there's different types of interactions that can drive this. One, hydrogen bonding. Two, ionic bonding. We've got amino acids that at neutral pH are positively or negatively charged. Third, we can actually get covalent bonds, which are known as disulfide bridges. And I'm not going to expect you to memorize all of these. I just want you to know that they exist right now. These disulfide bonds are created between two methionine amino acids, cysteine amino acids that both have sulfur in them. And then the last, hydrophobic interactions. Just like we talked about on Wednesday, how we can have oil and water and the oil kind of self-assembles into globs as a way to prevent its interaction or minimize its interaction with water. The same thing happens with proteins. Good example would be, I don't know if I have this one in here anymore. Nope, took it out. There are enzymes in our mouths, tears, etc., that are really strong antimicrobials. It's known as a lysozyme. Lysozyme is very hydrophilic. The outside is hydrophilic. But if the whole protein was hydrophilic, it would always lose its shape. So to help retain the three-dimensional form, the core of that protein is largely made up of hydrophobic amino acids that help to hold it all together. Okay? So there's a balance of both of these in proteins, and we'll kind of cover this a little bit more as we go along. But again, those polar interactions of covalent bonds are going to be what helps to contribute to activity here. Okay, I know we haven't covered all of these slides. I'm not really going to spend time talking about nucleic acids right now because we will spend a lot of time on that later on. Carbohydrates we will spend a bit of time on later on as well, so I don't want to spend too much now. Just a couple of things that I wanted to make you aware of. I'm going to go to Blackboard real quick to show these. All right. First one, the homework assignment for Monday, so this will be due Sunday night, if I'm not mistaken, is a file. Oh, it's not due Sunday night. You bring it with you. This is kind of like parts of a cell, of a typical prokaryotic cell. We're going to begin to cover some cellular components and talk about function. So print this out, or if you want to have it on your computer, that's fine. But what I'll have you do as soon as you come in on Monday is just show me your sheet of paper, or show me your computer to let me know you've got it. This one is a participation grade, okay? Not grading on correctness for this one. The second thing I wanted to point out was that one thing I am going to try to go to is for assignments like this last one, I will put together a brief little rubric for you. If you click on the link of the homework, you'll actually see now where it says how many points it's worth. There's a separate rubric, and it's a very simple rubric that I'll use throughout the semester to define points. And